how we can use transcriptome analysis to study evolutionary questions. And many evolutionary questions also overlap with questions in ecology. So I guess for those of you who come from more of an ecology background, this could also be interesting. And we'll be, the lecture will be in two parts. So first, in an hour or 1.5 hours, I'm not sure, will also depend on you, because we could try to do this interactively. We'll be studying a piece of transcriptome data. In fact, two pieces from two different studies. I joined them and study questions about primate testis evolution. Some of you might already be familiar with this, those from our group. Yeah, I think it's kind of an interesting case study. And then is testing a couple of hypotheses, observing a number of patterns. We will continue discussing theoretically how transcriptome data can be used to address evolutionary questions, and what kind of issues there are with study design, data collection, data processing, and what kind of analyses people perform. This will be pretty superficial, both because we are limited in time, but also because I'm also outdated now on transcriptome analysis. So my PhD and some of my postdoc work was focused on gene expression, comparative gene expression in humans and chimps and other primates mainly, but later I drifted away, and now most of my work is in ancient DNA and population genetics, and so I'm a bit rusted perhaps when it comes to comparative transcriptome issues. So I had to ask my younger colleagues to check if I'm not bullshitting. It seems like it wasn't too bad, but they had I had to have them check my slides. Okay, which are a bit outdated, as I said. But I guess, yeah, for the purposes of giving an idea about how we can use such data and what kind of issues might arise, it will be not really comprehensive, but some kind of informative or at least hopefully inspiring talk. Okay, our case study is primate testis transcriptome. So we're our closest living relative are chimpanzees, as you know. We differ in some respects, like the body hair, the brain size, but also the testis. So the brain size has been the focus of most evolutionary studies that compare humans with extant relatives, as this is an organ which has expanded in volume 3 to 4 times in our recent history, it appears. And of course, we're very much obsessed with our cognitive abilities, how superior we are, etc. And so a lot of work has gone into studying brain, how the brain evolved to be so large and function so differently, blah, blah. But as I mentioned, there are other aspects of anatomy that have changed as dramatically as the brain. And one such characteristic is the testis. So chimpanzees and bonobos, they have testicles that are 3 to 4 times the size of human testicles. With respect to other things like muscle strength, etc., there are also pretty dramatic differences. So in fact, it's not just humans and chimps, but if you look at gorillas, gorillas have even smaller testicles on average. So here you see on the y-axis testis weight, and macaques have slightly larger testicles than human and gorilla. But then if you normalize this by the body weight, which might be more fair because the species also differ quite a bit in their body sizes, then gorillas are dismally have dismally small testicles and macaques pretty large, etc. And this pattern was in fact studied, observed, and reported on already. Was that 40 years ago, nearly my age, by these guys, and they pointed out that when you study body weight versus testis weight, there is a pattern such that species which among primates, species which have multi-male breeding systems, or simply multi-male species. In fact, multi-male refers to not about males, but female behavior, and it's whether it describes whether a female is receptive to multiple males during estrus. So if she could basically get, if there is competition among males to get females pregnant by copulating more basically. So this is what's called a multi-male breeding system, or polyandry. And one alternative is monogamy, where males and females stick, at least during one breeding season, they choose one partner and go with that partner, and then there's no space for competition. Single male here refers to harem type of systems, polygyny, single male, multiple females. What is observed in gorillas, for instance, you have single male, multiple females, and then quite a few males who go without mating, bachelor males. And yeah, so if you study these two characteristics of these species with respect to also their mating patterns, you see that those, would you call that color? I don't know. Let's call it purple. Those purplish dots tend to be above the regression line, and blue and green dots, so those belonging to multi-male, monogamy. So basically single male mating types, they tend to be below the regression line. So this suggests that, so for one thing, we can conclude that these are all primates and pretty closely related ones. Chimps, by the way, are also multi-male, so that they live in multi-male groups of multiple males and females, and although there's some hierarchy where some males perhaps might end up mating more with females than others, still there is a chance that any chimp male can find a chance to mate. And so species like chimps versus humans versus gorillas, they seem to be differing a lot in their mating behavior. So this suggests that this behavior is changing pretty rapidly. Through what dynamics? I don't know, perhaps Erol, I also unfortunately missed Erol's talk, but perhaps he might have discussed models that were constructed to predict how mating type evolves. But by the way, Paulos came. Should I admit him? Okay, I'm not sure if I could anyway. So the question is whether okay. So the one observation is that this behavior seems to be very plastic, very flexible in evolutionary time, and it seems to be evolving quite a lot. And once a species moves to some equilibrium behavior of multi-male or single male breeding, its anatomy also seems to be following, such that irrespective of their relatedness, so these are not all closely related groups, and these neither, so This suggests some degree of convergent evolution of testis size of anatomy, given given the mating behavior. And indeed, as I mentioned, species like chimp and macaque, they're known for high female promiscuity, given female promiscuity, that creates space for competition among males, in sperm competition among males. And it's easy to conceive that that would drive large testicles and faster sperm, and also copulatory plugs in that would help a male prevent a second male successfully copulating, impregnating a female. Which are also seen, and at least they're seen in mice. I'm not sure if in chimps these existed. I'm actually not, perhaps in macaques, but at least in some mammals this is well documented. Some protein plugs that basically prevent the next male from fertilizing the female. And whereas in humans and gorillas, I mean, the current type of systems, of course, in humans, humans is a bit difficult to describe because, you know, we have too much behavioral flexibility, seems like. Our behavior is too much dominated by culture. And a culture is, you know, it evolves under all kinds of different processes, and so like it's kind of difficult to describe like a simple behavioral type for humans, but still, given the testis size body weight ratios, it kind of the pattern implies that at least our ancestors have been more single male like. Their behavior seems to be more single male like than multi male like. At least if we take the testis size as a proxy, and also I guess ethnographic studies would suggest there's more. I mean, we seem to be not as promiscuous as chimps. It looks like. And that means and in gorillas, where there's strict harem systems, as we mentioned, I mean, there again might be some, you know, escapes and exceptions, but in general, that's how they behave. So that means there's much less sperm competition among males, and it appears that this kind of evolving under these kind of conditions, that leads to smaller testes and slower sperm. Of course, the question is whether these attributes, smaller testes and slower sperm, whether, so imagine a species which is a lineage which is first multi-male mating and then switches to single male. The outcome, what we observe from the data, is smaller testicles, but is that because small testicles are advantageous? Is there positive selection for smaller testis size, or is it because you know, just random variants that would have been detrimental under a multi-male environment, in a multi-male behavior environment, variants that would be deleterious in multi-male systems, whether they become neutral, and then just if such variants arise frequent enough, they might just drift to fixation, and that might also lead to smaller testes just by genetic drift, mutation and drift. And the answer, I think, yet is unknown. I don't know, does any of you have an idea whether anybody tackled this question, whether this was positive selection or drift? I mean, you could also imagine perhaps advantages of having smaller testicles if there is a competition at some point, you know, perhaps easier to protect if an organ is smaller, but I really have no idea. Anyway, so this is the problem we're going to tackle with the transcriptome data. The main observation is that there seems to be some kind of convergent evolution. I mean, we say convergent evolution because I guess all of you here know well that, perhaps I can refer to the slide again. So the phylogeny of our species goes like this. Human and chimp are closest relatives, and gorilla is an outgroup, and macaque is an outgroup to all apes. And given that human and gorilla have very similar attributes, and chimp and macaque also have similar attributes, this seems to be a case of convergent evolution, or of course reversal. It could also be that the I mean, you could imagine either the, let me see, try to note here. So we could have a multi-male pattern here that was inherited to the macaque, and also multi-male pattern here that the chimp received, but then in the human branch and in the gorilla branch, there might have evolved a single male pattern, or we could also have a single male. No, let me see. So here we could have evolved a single male version. Well, yeah, I mean, it doesn't matter. So a single male, and then on the chimp branch, you might have multi-male. Yeah, this is, it's equivalent basically, right? So whatever you call reversal or convergent evolutions would depend on what kind of state you assume in at the ancestral ancestor, common ancestor of all these guys. And this is hard to guess, and it's even it would be even possible to assume that there might have been multiple changes also on the macaque lineage, for instance, given that even these very closely related species, you know, diverged within the last 8-9 million years or something. They show such divergence. So perhaps on the macaque lineage, 25 million years ago, there might have been multiple changes as well. Yeah. So coming back to our question, how transcriptomics can help study the evolution of these patterns. So we go back about 15 years ago, and there it was the time of microarrays still, but Afi microarrays, which were pretty reliable, not as noisy as earlier types. Does everybody know what a microarray is? A microchip. So some kind of tool to measure gene expression across thousands of genes, which was not possible before the basically 2000s. And these arrays, they opened up a totally new field of transcriptomics. And these guys, he happens to be early ex-PI of mine. He and his PIs, they studied gene expression in humans and chimpanzees in different tissues, as you see, 5 tissues. The trees reflect how much gene expression has diverged among individuals, and the distances indicate total divergence. So you see overall in the brain there's least divergence, and the branches that start with H are humans, those that start with C are chimps, and humans and chimps are really clustered. I mean, they do separate, as you see, but they're a small ball here in the brain, whereas in the liver and testes there's much more divergence. In the liver, everybody is very diverged, perhaps related to the liver being an organ which is pretty responsive to the environment, so it might not reflect phylogeny as strongly as some other species. And then testis, what was dramatic was within species, as you see, within species, diversity is pretty small, but between species, there's a lot of divergence, much more than other tissues. And in this paper, they were also showing that testis-related genes show more evidence for rapid evolution than genes expressed in other tissues. And thus they were concluding that there must have been a lot of, instead of the brain itself, the testis might have undergone a lot of expression evolution driven by positive selection. On the chimp branch or on the human branch, can they tell, looking at this pattern? They can't tell, right? What would you need to drift hypothesis? You did not make your slide full screen. Yes, I know. I just want to follow what I'll be talking about next slide. The drift hypothesis basically, sorry, Pavlos, coming back to this. So what we see is in the tree, so assuming the ancestral pattern is multi-male, and chimp also acquired the ancestral pattern, and then in the human lineage and in the gorilla lineage, there evolved a single male pattern, single male behavior, and along with that, the testis sizes in gorilla and human reduced. And the question is this reduction? So we could imagine perhaps when large testicles are not necessary anymore, and if they're also advantageous with respect to protection, for instance, or if they bring about a cost, then they might they might reduce in size through positive selection, or alternatively, if there is no cost to having large testicles, or no very strong cost, if there's no strong selection against large testicles in the single male environment, then still, if we can imagine that variants that affect the testis phenotype that have a reducing effect, if they occur just simply more frequently, deletions, as far as I know, that's deletions do tend to occur more frequently, at least disruptive. I mean, it's obvious that disruptive mutation, it's easier to have a disruptive mutation than a mutation than how they call it, like a gain of function or overexpression mutation. I think that's pretty established. I'm not sure if there's like a systematic study about this, but I guess it makes intuitive sense that disruption is easier than disruption, and reduction expression is easier than overexpression. If so, by mutation, by the accumulation of mutations with such disruptive effects on these two lineages, we might have ended up with smaller testicles. The difference would be, how could we potentially test this? If we can identify those responsible variants, which could be numerous, of course, so most likely some polygenic trait, we could try to see if there is we could try to see selection signatures, if they exist or not. But of course, selection signatures, especially if they arose long time ago, might have been lost, so their lack wouldn't really tell much. Yeah, it wouldn't be easy. If we had ancient DNA from like Australopithecus or something, which is not possible, but then we could at least study past the rate of accumulation of such variants, and that could give us an idea about whether it's selection or drift. But otherwise, I'm really not sure how we could distinguish between the two hypotheses. Pavlos, would you have a suggestion? Is Pavlos there? Or anyone else? How could we compare these two hypotheses? Not real. Okay. Yeah. Well, anyway. Oops. Now I cannot. I made the point there. Okay. Coming back to our transcriptome thing. So this was the observation. A lot of divergence between human and chimp. Yeah. Again, we don't know which species diverge. For that, we would need what? Anybody wants to contribute? We would need yeah. How could we determine whether the divergence here between species is divergence on the human lineage or the chimp lineage? Madame the Monsieur. How? We would need an outgroup, right? If we can use gorilla as an outgroup, if gorilla is a good outgroup, that could be also debated, but then we could decide if gorilla behaves like human, then we could decide that changes have occurred on the chimp lineage. If gorilla is more like the chimp, then we could assume it's the human lineage, or if gorilla is in the middle, then we could say both species are diverged. Okay, but in this study, they didn't have a gorilla. And then a later study from 10 years ago now, this was one of the first RNA sequencing studies to investigate transcriptome patterns across different mammalian organs. And in the testis, they were noting this, this pattern that they built a tree of gene expression. They also had mouse and other species, as you see. And there they were seeing that gorilla, they had one gorilla male, and gorilla seemed to be closer to the human than the bonobo and macaque. And that, of course, supports the idea that indeed this could be change. So the chimp might have diverged more. And this was interesting, but these guys didn't really do much further on this topic. So one thing we had, we decided to do a long time ago, and finally now we've managed to finish up the paper, and it's under revision anyway. So we decided to join these two data sets where we would have a lot of humans and chimps, and also gorilla and macaque and other species, and then test if there is a convergent evolution in testis transcriptome levels. So if depending on the mating type, species appear more similar than you would expect from their phylogenetic relationships. Because normally, if for an average gene that is not being that is neutrally evolving, let's say, in this expression, we would expect gorilla to be equally distant to human and chimp, right? Is everybody okay with that? On average, gorilla should be, in its DNA sequence, gorilla is equally distant to human and chimp. For an average neutrally evolving gene, could there be such, like, take a pseudogene, and everything else being equal, the gorilla should, on average, be equally distant to human and chimp. And macaque should be equally distant to all of these, right? Given the phylogeny. Everybody can read the phylogenetic trees. And if so, we can ask, does the expression patterns in the testis also follow this, this expectation, or do they diverge? And the first goal was to combine these data so we have more power to see what's going on. We don't have to rely on just two humans and two chimps. And that is what we will now start doing. Combining two, let me share my desktop. I think this is easier. You see my desktop, right? Hopefully.
Yes, we can.
Yes, okay. So the data, one is microarray data, one is RNA-seq data. Can we really join them? Who thinks we can join them? If we had time, I would have run a poll, but anyway, what's your feeling? Okay, somebody has, Maria says no. answer is both no and yes. So, well, we'll see what we can do. But I got from my colleague the two data sets. I load them. So many of you are already familiar with R. I'll also share with you this code. And the data is, the microarray data is an AFI data. So it's, I think this was the name. Looks like this. The humans, HT for human testis, CT for chimp testis, and these are ensemble genes and gene expression levels. How big is it? Let's check. About 19,000 genes measured on the microarray. This is a microarray. It's been processed in slightly special ways such that the probes that do not match between human and chimp, they've been computationally removed, so the chip behaves fairly to chimps. The chip is fair to chimps, yes. Okay, and the other data set, very creatively called bigger. I'm not sure what they call bigger. Anyway, it looks like this. So we have a different, it's not in a perfect format as many times in real life. That's what you get. Somebody compiled it with different gene IDs, chimpanzee ensemble gene IDs, macaque, this is human ensemble gene IDs. And then this is GGOs is for gorilla, the two humans, two macaques, and this is Pan-paniscus, bonobo, pan-troglodytes, this is a chimp. Common chimp. And we'll treat bonobo and chimp as the same species. They're anyway very close by. Okay, so somehow we now need to join these two data sets, and so it's a bit of an R exercise. I just renamed the two data sets, MAT1 and MAT2 for simplicity. So I renamed the thing, and yeah, we already checked the, and as you see, this is the normal ensemble gene IDs. These are ensemble gene IDs, but with a suffix, which we should remove. Where that suffix comes from, let's not discuss for the moment. So we can remove this part, and also for simplicity, we can remove this dot cell part. So how can we do that? In R, there's this nice G sub function. And it recognizes a pattern and it replaces it with some other pattern. Here, I just want to remove it, so I can use G sub as like this. And here, if you have any questions about any of the commands, please feel free to ask. By sound, it's probably better. So what I did, as you see, I removed the underscore ATs. Okay, and let me also remove the dot cells just for the dot cell is for visual purposes. I don't want to deal with long column names. And now the other data set, it needs a bit more processing. Apparently, there are also some. I wanted to have column that. Okay, let me show you that. I haven't defined MAT2 yet. Yeah, so this is my matrix. I actually want to get rid of these columns. I want to assign this column as row names, and then I can use those common row names to join the two matrices. But it turns out, I mean, I'm not showing it to you, but if you try to do that, there's an issue because there are some ensemble gene IDs duplicated multiple times in this few, but some cases, and then you can't assign multiple row name, the same character string as row names to multiple rows in R. So I get rid of the duplicated genes, and then I assign row names to my matrix. So now it looks like this. Okay, now I have my row names here, and the last step is to get rid of these 1, 2, 3, 4, 5 columns. I do that. Check my matrix again. Sorry, here doesn't work because this are marked down. Anyway, okay, in format I need, and let me also check the dimensions. Okay, 7,000 genes only here. In the study, they were only including genes with one-to-one orthologs across these species, especially because macaque is a bit distant. Of course, but we ended up losing genes. I'm not sure if there was other filtering involved. Anyway, so we have these two matrices. They both have ensemble gene IDs in their row names, and we can use this merge function in R, where you can merge two matrices by their common columns or by their row names. And here they have row names in common, so I can run this and check. Sorry, this doesn't work. Again, yes. So my resulting matrix is contains now the Kaitovich data, the microarray data, and then still the microarray data for chimp, and then this is the RNA-seq data. Did I tell you this was RNA-seq? Yeah, I hope I did. So one is microarray, one is RNA-seq. One is 20 years old, one is 10 years old. Okay, and so we want to continue working. So one thing we can do, for instance, is to construct a principal components analysis to study the overall variation in gene expression among these within this matrix. And we can do that, but first we need to get rid of this column because this is non-numeric, as you see. So one thing, and it's also nice to have the row names back again. So what we can do is we can assign row names first as the first column. So the first column I take and assign the matrices row names with that. And then I remove the first column itself, and then okay, now it's in nicer format. Let me also check, this is a data frame. So data frame can be dangerous because sometimes it looks as if it's containing new numbers, but those numbers might have become factors, and then everything gets screwed up. So just to make sure, we can apply okay, everything is numeric. I hope everybody knows this apply function. It applies some function in R2 means across the columns. So I check the class of each of the columns, and this is fine. They're all numeric. What's the dimensions? Okay, 7,000 something. That's under. So initially in the smaller matrix, we had 7,700 genes, and now about 400 genes we've lost. That's kind of normal. Okay, and now let's see if okay, we did join the data, but does the data make sense? Does the joint data make sense? And for that, I'll be doing a principal components analysis. And also, okay, actually before that, what I was doing here was first study the general expression patterns in the distribution of expression levels across all the columns. And for that, here I'm not sure why I was converting into matrix, but it doesn't matter. Let's do that. So we have the same thing. And if we then draw a box plot, my box plot is somewhere here. So what do you see? The distribution, these are the microarray data, log transformed and normalized. And this is the RNA-seq data. As you see, the the scales are super different, right? On average, they're all kind of similar, but the scales are super different. And that already hints that probably this won't really work, but still, let's continue doing running a principal components analysis. And if we do that, how do you think the data will cluster by? What will principal component 1 be correlated with? You should be able to guess this if you're familiar with some computational analysis. What would principal component 1, PC1 be correlated with in this data? The species, the mating type, or something else? Come on. What will principal component 1 be correlated with? Species IDs, mating behavior, or something else? Come on, it shouldn't be so difficult. Maria, when you said no, what did you have in mind? Data type. Okay, assay type. Yes. Okay. Thank you. Yes. Data type. So that's our guess. Given that it might have so strong impact on the data. So the one way to run principal components in R is as follows. You need to transpose the matrix because it calculates components across the rows instead of the columns. And the scale true, which means each row should be, was it row or column? I forgot. Anyway, it's scaled so that each component, yeah, in this case, each column it should have equal mean and variance so that they're all contributing equally to the overall variance. All the genes are contributing equally. And we run this, and then PC, so if you just say PC, you will obtain a lot of results. That's for somebody PC. Yeah, so here you get the how much, what proportion of the total variance is explained by PC1, PC2, etc. It's in diminishing order, as you see. And if you plot the first two PCs against each other, so this PC object has this sub-object X, which are the values of the PCs for each entry. And then if you plot them, oops, yeah, I had also described a columns vector. Here I was using grep, as you see. I hope you can easily see. Yeah, I was using grep. So for instance, what I did was I got the column names of my matrix. I want to convert humans, these into the species IDs into colors. And what I did was I grabbed H, which is only found in humans, and replaced that with red. So basically it went like this. So this was colors first, and then I grabbed H, and then replace those elements by red. So this returns me an index vector. And then choosing those elements, I replace humans with red, and then chimps have either P or C. I'm lucky that there wasn't a diversity or other similar species names. I replace those with blue, blah, blah. So in the end, I get a vector of colors in the same order as my columns, as the columns in this joint matrix. Now I can use that to plot my, here you go. So human, gorilla, chimp, and macaque. But this is the RNA-seq data set, and these are all the microarray data sets. I can also add the names of the, oops, sorry. I can add the names of the guys, and as you see, there's the only real clustering seems to be in the first PC. I also checked the third and fourth PC, also doesn't look much better. Yeah, and we can also see what's going on just studying a single random gene. Here I chose the third gene of this matrix, and okay, sorry, I should have shown you this. Yeah, this is the expression level of the first gene, sorry, third gene, and the index is just random, the order of the individuals. These are humans, blue are chimps, gorilla, human, chimps, macaque. And as you see, in fact, there is a trend of higher expression of humans than chimps in both cases. If we zoom in the microarray part of the data for this gene, we see a pattern like this: humans, chimps. This is significant, right? I mean, any test, parametric, non-parametric, would tell you this is significant. And then this is the RNA-seq data. In the RNA-seq data, we also see the same thing: humans here, chimps here, macaques kind of closer to chimps, and gorilla above. So, in fact, very much following the behavior, mating type behavior pattern. But because won't we transform the matrix before applying? Yes. Well, yes. Wait. Transform, you mean, Maria, was when you say transform, normalize to remove this batch effect? Was that your point? If that's your question, yes. If your question is about transposing, then we did already transpose. But transposing and just scaling the matrix, this is already log transform. This is already log transform data. So expression values usually their range is much larger, and this is if you already see data points like this. Where's my box plot? My box plot. Yeah. So if you see a range like this, this is log transform data, for both of them. Microarray, both microarrays and RNA-seq. Microarray, one thing to keep in mind, microarrays are relative expression levels, so that it's just some intensity level relative to the background, which is interpreted as gene expression. Okay, it's not absolute. Here we have absolute counts assigned to transcripts. So we have, yeah, it's just different, much more direct raw information here about gene expression. This is only indirect information. So it's really like, in a sense, in terms of data type information source, it's apples and oranges. Yeah, but okay, let me just finish this part. So basically, for this gene, what I was telling you, in fact, there is a consistent effect between the two data sets, humans or single male species having higher expression than multi-male species. But because we join them together, we basically don't see much. So what can we do? Okay, let's continue discussing this after the 10-minute break by, well, here by 4 p.m. Okay, and you can already think about